Okay, I think we're a couple of minutes past 4 o'clock. So we'll make a start. Thank you for joining us. Welcome to this UK Dementia Research Institute or DRI research webinar on Huntington's disease. Please note that it is being recorded. I'm Tom Massey and I'm chairing this session. I'm an MRC clinician scientist and DRI group leader here in Cardiff, where I co-lead the Huntington's Disease Centre in Wales. It's a great pleasure to welcome Sarah and Gabby to speak today, and I'll introduce them properly in a moment. First, I've got just a couple of slides on the UK DRI for those of you who may not be familiar with it. If we have the next slide, please. So the UK DRI was founded in 2017 by our principal funder, the Medical Research Council, and the two dementia charities Alzheimer's Society and Alzheimer's Research UK. And we're now fortunate to receive support from a number of organisations, including LifeArc, British Heart Foundation, NIHR and Parkinson's UK. And the UK DRI vision for the future is summed up by this phrase, which is healthy brain ageing for all. At the heart of the UK DRI's work is unpicking the molecular mechanisms that lead to neurodegeneration. And by understanding the causes and drivers of disease, we're all aiming to develop new diagnostics, technologies and treatments so that can benefit all those people affected by or at risk of all forms of dementia, including Huntington's disease. Since being founded in 2017, the UK DRI has grown to 9 research centres hosted by leading UK universities, and there are now 85 labs of group and emerging leaders and over 1,000 people, researchers, clinicians, students, support staff, working together to try to transform the lives of people affected by neurodegeneration. The next slide, please. So we'll now move on to the webinar itself. This is the third instalment in this UK DRI research webinar series called Rethinking Neurodegeneration. And today, Professors Sarah Tabrizi and Gabriel Balmus will discuss the question, could DNA repair hold the key to treating Huntington's disease? So our speakers are firstly Professor Sarah Tabrizi from UK DRI at UCL in London. Sarah leads a bench-to-bedside research team focused on understanding mechanism, validating targets, and finding disease-modifying therapies for Huntington's disease. She co-founded the UCL Huntington's Disease Centre in 2017 and helped set up the UK All-Party Parliamentary Group for Huntington's disease. Most recently, Sarah has served as the lead scientific advisor for uniQure's phase 1/2 clinical trial for AMT-130, which is the first gene therapy to be tested in people with Huntington's disease. In the UK, this was delivered by Intricate Neurosurgery here in Cardiff by Professor Liam Gray and his team. And really excitingly, as you may have seen last week in the news, top-line data from the company indicated that treated patients had significantly slower disease progression than matched controls. And this is the first time disease slowing has been seen in an HD trial, which is amazing. Sarah's achievements have been recognised with numerous major prizes. And she's been elected Fellow of the Royal Society, UK Academy of Medical Sciences, and the US National Academy of Medicine, amongst many others. Our second speaker is Professor Gabriel Balmus. Gabby joined the UK DRI at Cambridge in 2018, interested in mechanisms controlling the maintenance of nuclear and mitochondrial genomes in mature neurons. He got his PhD in 2013 from Cornell in the USA, and went on to complete postdoctoral training at the Gurdon Institute in Cambridge and the Wellcome Trust Sanger Institute. His work has revealed critical mechanisms by which DNA repair proteins like FAN1 either protect against or promote toxic repeat expansions in Huntington's and related disorders. And this is opening up new therapeutic avenues. As UK DRI group leader, Gabby brings his wealth of expertise of research in genome instability to neurodegenerative disorders. So to date, Sarah and Gabby will be discussing evidence for the role of DNA repair in Huntington's disease, their collaboration in this space, and also the future of therapeutics for Huntington's disease. Please put your questions, very importantly, if you have any questions, put them in the Q&A box throughout the talk, and then we'll try and answer as many as possible, or at least Sarah and Gabby will try to answer as many as possible after their presentations. Okay, thanks. So I'll hand over now to Sarah, who's going to speak first, I think. Sarah.
Thank you so much, Tom, for that very kind introduction. Thanks to DRI for inviting me and Gabby, and it's a delight to be able to talk to you today about DNA repair, which is something Gabby and I work on together. And we met through the DRI, and we have a wonderful working relationship and collaboration. So Huntington's disease is a dreadfully devastating neurodegenerative condition. This is a lady aged 38, and this is her 13 years later. It's a lady from Lake Maracaibo in Venezuela with end-stage disease. It affects people in the prime of life. It's fatal, it's monogenic, it's fully penetrant, and we currently, until last week, actually through a few weeks ago, have no evidence of disease-modifying treatments. It's caused by a repeat expansion in the first exon of the Huntington gene. And if you inherit 40 or more repeats, you always will develop the disease if you live long enough. It's fully penetrant. And it encodes it's a huge gene. It encodes a protein, mutant Huntington, that has a number of mechanisms of pathogenesis. But we really understand the mechanisms of the pathogenesis now in a deep way. So Huntington's genetics, if you have 40 or more repeats, you always get Huntington's disease. And there is a CAG repeat threshold with a reduced penetrance range. Between 29 and 35 is the intermediate range, which doesn't cause disease to the person, but can expand into the disease range in future generations. So what we understand now about the molecular basis of Huntington's disease is that it's in two steps. The first step is the rate driver of disease, and we now know that that is somatic expansion of the CAG repeat. So you're born with, say, 42 repeats, which is the commonest repeat, and that's in your blood. But we know in your brain that that CAG repeat gets larger and larger. And that was first reported by Peggy Shelbourne and colleagues in the early 2000s. And no one really understood the relevance of this data. This was post-mortem data with a southern blot, and there were repeats up into the high hundreds. But that was a very good observation by Peggy early on, and we now know, and at the time it was pretty much ignored, but now we know that the somatic CAG expansion is what is driving the disease and the rate of somatic expansion in the brain. And that became a really big area when in 2015, from 2015 onwards, a number of genetic modifiers in DNA damage repair pathways were identified that modified the age of onset. And in our lab, we identified MSH3 as a genetic modifier in 2017. And I've been working on it since then. So what we now know is step one is the rate of your somatic repeat expansion in your brain drives Huntington's age at motor onset and rate of progression. Step two is the toxicity driver, and that is at the protein level. So the Huntington gene is transcribed into the full-length Huntington mRNA and also an alternatively spliced exon 1 Huntington 1A mRNA, which was described by my co-director here at the UCL HD Center, Gillian Bates. When the repeat gets larger, more of the toxic exon 1 is produced. And in our lab, we've looked in human medium spinal neurons, and when the repeat gets above 140, about 8% of the transcript being produced is the Huntington 1A fragment. So toxicity, the full-length mutant Huntington undergoes N-terminal proteolysis, and the proteolytic fragments cause toxicity, as well as the highly toxic exon 1 protein that forms nuclear and cytosolic aggregates. And the therapy that Tom mentioned early in the talk, the microRNA gene therapy, actually targets this species and this species. It targets the full-length mutant Huntington mRNA and the Huntington 1A mRNA. There are many pathogenic cellular mechanisms in Huntington's disease, like other proteinopathies, There is aggregation, impairment of proteostasis, synaptic dysfunction, axonal transport impairment, nuclear aggregation, and very early dysregulated transcription. And repeat expansions, Huntington's disease is one of many repeat expansion diseases, and I think they're hugely interesting. They all affect the CNS, and they represent a significant unmet need. There are over 50 that are known, and many of you will know Fragile X, Friedreich's, myotonic dystrophy, the spinocerebellar ataxias, and a very nice paper last year from Ariana Tucci here at UCL. She published in Nature Medicine whole genome sequencing of nearly 83,000 people. Overall allele frequency of 16 repeat expansion diseases was 1 in 283. The commonest was C9 at 1 in 839, myotonic dystrophy 1 in 1,786, Kennedy's disease 1 in 2,561, and Huntington's disease was 1 in 4,109, much more common than previously thought. And the reason I mention this, all of these repeat expansion diseases exhibit somatic repeat expansion in affected tissues. And I think that all repeat expansion diseases will be shown to have a similar mechanism to Huntington's disease, somatic repeat expansion and then toxicity drivers. So why do I think targeting somatic CAG is so important? Well, you're born with the mutant Huntington gene, but we know it expands in the brain. We and others have shown that the more expansion you have in the brain, the earlier onset, and those with less expansion, their later onset. And that can be modified. Your rate of somatic expansion is modified by now multiple mismatch repair genes. And so they're all associated, all these genes are associated with altered age's onset and progression. And MSH3 was the one that we described in 2017, and now is a very big area of therapeutic interest. This was the paper, Davina Hensman-Moss in my lab. She published part of her PhD, the MSH3 signal. And as I say, since then, Gabby and I have been working on this. We found, Mike Flower in my lab found that if you had the MSH3 polymorphism, you actually had less MSH3 at the protein level. So it was working through a loss of MSH3 function. And many of you will know David Altshuler. He's the Chief Scientific Officer of Vertex. And David wrote this beautiful Nature Reviews Drug Discovery that if you go for therapeutic targets that have been validated through human genetics, they have a much better chance of working. So human genetics and Mother Nature has actually done the experiment with MSH3 and PMS1 in that those people who inherited polymorphisms that downregulate MSH3 or PMS1 have slower disease progression. And those who have inherited polymorphisms that actually upregulate FAN1, another DNA repair gene, have slower progression. So these are hot targets in Huntington's disease. So how do we think this is happening? And this is a very nice paper from Vanessa Wheeler in Nature Genetics this year. And I think she simply shows what we think is happening at the repeat. So in the repeat, the long CAG is transcribed, but during transcription, these loop outs occur. This is quite simple, but this is the mechanism. And these CAG slip outs occur, and they are recognized by this complex MUT-S beta, which is MSH2 and MSH3. MUT-S beta recognizes the loop outs, then recruits MUT-L gamma, which is MLH1 and MLH3, and MUT-L beta binding. And that process results in MUT-L gamma nicking the opposite DNA strand, and that nicking results in this DNA synthesis with Pol gamma, and the new DNA synthesis results in a lot longer expansion. So this is a very simple model of a very complex mechanism. And because this is a short talk, I would love to go into more detail exactly what's happening at the slip outs and the mechanism of action, because it's what a lot of my lab is working on. But basically, MUT-S beta binds to the slip out, recruits MUT-L gamma and other MUT-Ls. It then nicks the DNA, and the DNA polymerases resynthesize the DNA, and that resynthesis causes an expansion. So that's simply in a nutshell. So what we want to do is target that process. And I'm just going to give you a few recent advances that came out this year. One was a very nice paper in Cell from Steve McCarroll's group, where they had done single nuclei CAG repeat sizing, and Steve developed a very nice method of sizing the CAG in single neurons. And he studied the striatum and found that in post-mortem tissue, there was a wide repeat length distribution. And there was many of the repeats in the between 40 and about 90, and they were relatively stable. And then there was these longer repeats right up to several hundred in neurons. And so Steve and Bob Hensecker used the data to put together a computational model. In that you're born, say, with 42 repeats, which is the commonest repeat. It expands very, very slowly. Once it gets to about 80, there's accelerated expansion. And then when it gets to about 150, there's widespread transcriptional disruption. And then soon after that, neurons die and collapse. And we've been modeling this in the lab and find indeed it's slow until you get to about 110 repeats. And then the repeat expansion is exponential. And we're trying to model in the lab what Steve was doing with postmortem brain. We also published a paper, Steve and our paper came out virtually the same day, showing that repeat expansion in blood was associated with biomarkers of neurodegeneration in Huntington's disease brain decades before people show signs of the disease. And so Steve's data supported that somatic expansion was driving the disease early on, and our data supported that somatic expansion was driving the disease early on in living humans. Steve's data was in post-mortem samples, and they really complemented each other. So what we found using measures of somatic expansion, we studied people decades before symptom onset and used causal modeling approaches to try and predict what somatic expansion measures predicted. And what we found is that somatic expansion in blood DNA is detectable and occurring as early as stage 0 and 1, which is decades before people show signs of the disease. We found, this is in subjects who are stage 0, nothing to find, completely well, stage 1, stage 2, that there was a significant change in somatic expansion even 25 years before onset. And this is over time, over 5 years, you can see the rate of somatic expansion in blood. And we looked at that with repeat. The longer the repeat, the higher the rate of somatic expansion. We then looked, and your rate of progression of somatic expansion predicted the earliest loss in the caudate nucleus. It also predicted the earliest loss in the putamen, key structures affected in Huntington's disease. And we found that your somatic expansion was strongly predictive of losing neurons in the brain as measured by CSF neurofilament. And this is people decades before onset. And so this was also looking at somatic expansion. We developed a measure with Henrik Zetterberg of indirect medium spiny neurons, which are affected very early in Huntington's disease. And that measure was proencephalin, which is an indirect medium spiny neuron neurotransmitter. And with Henrik in the DRI, we developed this novel assay, and we found that your somatic expansion rate predicted how quickly you would lose indirect medium spiny neurons in the striatum, really tying somatic expansion to the fundamental early processes in this disease. And so what we did is put forward this model where you're born with this inherited repeat length, somatic expansion is occurring in your brain, and also we can measure it directly in your blood because we can't do brain biopsies in living young people. It's modified by DNA mismatch repair, and we can use measures of somatic expansion in blood to predict what's going on in the brain. And we found it predicted neurofilament increases, proenclephalin loss of indirect medium spiny neurons, and the earliest caudate and putamen striatal loss in stage 0 and stage 1, decades before onset, really reinforcing that somatic expansion is driving the rate of this disease. There's a very nice paper as well by William Yang's group in Cell, which showed that mismatch repair genes drive the pathogenesis in mice. And when he knocked out either MSH3 or PMS1, he corrected synaptic deficits, astrocytic deficits, and locomotor defects in HD mice, showing that MSH3 in mice could be modulated and it changed the trajectory of the disease. So MSH3 is an ideal therapeutic target. We did this analysis showing that MSH3 loss of function is tolerated in humans from the exome sequencing data. You can see here observed versus expected loss of function. And see here with MSH3, above 0.6 suggests the gene knockout is tolerated, and this is well above that. So MSH3 and PMS1 looks safe as a target. It also affects multiple repeat expansion diseases, so it's potentially could treat not just Huntington's disease. And if you knock out MSH3 in mice, they're fine, they don't develop tumors. And so we developed an MSH3 antisense oligonucleotide, and we published this paper in Science Translational Medicine earlier this year with Gabby and I. And we found that MSH3 ASO slowed somatic expansion in our striatal-enriched human neurons. And you can see here there's a dose-dependent slowing of somatic expansion with the MSH3 oligo. And we found that when we were looking at IC50, that if you inhibit somatic expansion with MSH3, 41% lowering of MSH3 halved the rate of somatic expansion, and 83% lowering of MSH3 stopped somatic expansion. And in knockout of MSH3, there was a tendency to contractions. So this oligo is now moving forward for clinical development, and it's a big area in HD. So what's the best time to treat for different therapies? What do I think? Well, this is the HD integrated staging system which we developed, which really stages Huntington's into 4 stages. Stage 0 to stage 3. Stage 0, you're born with the gene, nothing to find. Stage 1, you're completely well, but you've already begun to lose some volume in your striatum, putamen and caudate. And then stage 2 is the earliest symptoms, very earliest symptoms. And then stage 3 is when you're beginning to lose function. So I think therapies targeting somatic expansion should absolutely be in stage 0 and stage 1. That's when I think somatic expansion is really driving the pathogenesis. And then therapies targeting the protein toxicity, I would say in late stage 1 and in stage 2 and potentially early stage 3. And the gene therapy results that I announced last Wednesday, the group that were treated with that is in late stage 2, early stage 3. Again, a Huntington lowering. So I'm now going to hand over to Gabby, who's going to talk to you about how we are trying to target DNA repair as a treatment for HD and other repeat expansion disorders. Over to you, Gabby.
Thank you, Sarah. Let me share my screen. There we go. Share. Can you see my screen?
Yes.
Perfect. Thank you. Let me just see the zoom also, because then I need to look at someone. Maybe now. Okay, laser, here we go. Can you see my cursor?
Yes, we can. Yeah.
Okay, perfect. So what I want to take over from Sarah and tell you a little bit on how I think about Huntington disease and how we might drive therapeutic finding through functional genetics. And I want to start where Sarah left, is this HD staging. And I think this staging is of critical importance. This was done by Sarah in 2022. And until then, the staging of Huntington disease was not done. And what Sarah nicely done kind of show how the disease progresses. And this is quite critical for clinical trials. And what she also showed that depending on the somatic expansion of CAGs in the neurons, this staging also changes. So if you have a shorter CAG repeat, then the latency to the stage 3 is later, and if you have a higher CAG, this happens faster. And because I came into neurodegeneration from cancer, I thought this was such a neat thing to do, because that's what actually people are doing in cancer, and that where success came from in cancer. The same kind of staging system has been used in cancer for the past decades. And what you see here on the right is actually how cancer survival rates over time increase with research from 1950, where the 5-year survival time was below 30% of patients, where now we're about 70%. And this success actually came in from understanding the staging of cancer and doing combinational therapy for different type of cancer. And I think really it's this bespoke combination therapy and targeted therapy that is success in cancer. And why bring cancer? I bring it because cancer and neurodegeneration actually are on the same balance at opposite ends. They both control cellular homeostasis. In cancer, you have increased cell proliferation that escapes control, and you have uncontrolled cell proliferation. While in Huntington disease and other neurodegenerations, you lose cells in neurons. And these are side by side. I lost my cursor. These are side by side with the same direction DNA repair. So in cancer, most often you lose DNA repair proteins, and that actually creates cancer cells sensitivity to certain chemotherapeutics. While in Huntington disease, actually you gain capacity, you have increased capacity to repair through mismatch repair, and that promotes this acute repeat expansion. And when you think about how these factors work into cells, you kind of think that there are thousands and thousands of genes that interact. And cells are actually built on proteins that are coded by genes that are organized in networks. And how do you make sense of all these modifications? Because if you think about one gene modifying in one part of the cell, that might modify things very further away, and you don't know where you can attack the disease. And when I think about this, I think of two evolutionary researchers. One is Jean-Baptiste Lamarck, and he had this idea of intrinsic evolution, and that evolution occurs within individual, changes are directed by environment, and acquired traits are inherited. The analogy here are neurons, where pathway rewiring happens in the neurons during the lifetime of that neuron. While the extrinsic evolution is that occurs across population, that's Darwin review of evolution, and changes are directed by variation. Favorable traits are selected by clonal selection in cancer cells. So cancer cells divide many times, and then they have a selection. So when you think about these two concepts in no proliferation and proliferation, you have to think that as CAG accumulates and Huntington protein accumulates, the neuron start to rewire its circuits either through epigenetic drift, stress-induced transcriptional programs, and accumulate these new proteins that define the cell trajectory. While in cancer, for example, all of this relies on heritable variation, and then you decide how you might kill cancer cells. So it's really important to understand how these pathways work, so that you can make sense or work to attack the different new generation or cancer in that case. And these principles came in cancer through understanding this rewiring. So normally in a normal cell on the left, these are two healthy cells that have different genetic pathways. And if you mutate a gene A, for example, that might become a cancer cell and it might proliferate aberrantly. But then the cells actually start to rely on another gene called gene E, for example, and that makes that gene escape killing. But then if you inhibit gene E, then you kill the cancer cells while the healthy cell is spared. And this is the principle that have been coined by my postdoc supervisor, Steve Jackson, where he showed that by inhibiting PARP in BRCA1 deficient cancer, you can specifically kill cancer cells, and that term is called synthetic lethality. So can we apply similar genetic concepts to Huntington's? And what, as Sarah said, in Huntington, the normal neuron has a CAG slower than 36, and that's where the mismatch repair pathway, the DNA repair, just goes on and screens the Huntington locus. And of course, because there is no expansion, there is no hairpins from those that Sarah showed, then the cell is left alone and continues the normal life. When you have a mutated Huntington, when you have this CAG repeat expansion depicted here on the right, what you see is expansion of these CAGs in the neurons, and eventually you also get accumulation of this mutant Huntington that kind of creates transcriptional havoc. And what you want to understand is how this happens in the brain. And I'm coming back to this cartoon that has been shown by a preview by Jill Bates on Steve McCarroll's paper. And you have to realize that in the brain of a Huntington patient, in the same time, you have different cells that have different sizes of CAG repeats. Each dot in here graph represents one neuron. So some neurons will have CAG repeat between 30 and 50, and others will have over 100, 200, 300. And it's between these phases where neurons start to drift and lead to cell death. And that's what both Steve McCarroll and in here and Nathaniel Heinz have shown, that if you take single cells from these brains and you look at them one by one, you can see that not all the neurons have the same CAG expansion. Actually, what you can see that many neurons have different sizes. So in the same brain of the same patient, you have neurons that have different sizes of repeats. And when you look at these neurons that have increased repeat expansion and you do analysis of transcriptome analysis, you can see that there are thousands of genes that are being modified when you have more than 150, when you have this transcriptional dysregulation. And then to understand better the pathway in terms of DNA repair, others like Vanessa Wheeler in here started to look at other DNA repair genes, and they have shown that MSH3, MLH1, PMS1, and other genes depicted in this Nature Genetics paper also modify repeat expansion. Moreover, William Young, as Sarah also showed that if you delete MSH3, you actually rescue the Huntington pathology in a mouse model of Huntington disease. And this has been shown also by Sarah and us in neurons, where if you inhibit MSH3 in iPS-derived median spinal neurons, you also stop repeat expansion. Furthermore, MSH3 doesn't work by itself, and we and others have shown that FUN1 is another protein involved in this mismatch repair recognition and helping mismatch repair. And FUN1 is an exoendonuclease, and here Chris Pearson has shown that both the exo and endonucleases are important for FUN1 function, and that Anna Klunicek also showed that PCNA is required for FUN1 interaction, so to allow FUN1 to go the damaged sites. And together with Alex Sartori and another paper from us and Sarah have shown that actually FUN1 and MLH1 and MSH3 interact to allow for proper DNA repair. So what's happening in the disease is that you have this initial step that Sarah also mentioned, where it's mainly based on this repeat expansion. And this repeat expansion is driven by erroneous DNA repair that is conducted by MSH3 and other members of DNA repair. Once neurons start to expand and have addition of many CAG repeats, about 150 and 160, you have this transcriptional disruption and accumulation of the mutant Huntington that then starts to create a lot of problems in the cells. And you have shown from the McCarroll paper that there are thousands of genes that are deregulated once mutant Huntington starts to accumulate. And so the therapeutic opportunity here would be that in the early stage, you'd want to inhibit MSH3 because that will stop repeat expansion, will not allow the accumulation of mutant Huntington. In a later stage, you probably would want to start with mutant Huntington lowering, but also on top of MSH3 inhibition, because as I mentioned earlier, not all the neurons are synchronized in a CAG repeat length. So some neurons in a patient will have smaller sizes while where expansion still happens, while others will be more towards the mutant Huntington toxicity. Actually, what I think one should do is actually try to slice the disease even further to understand gene-gene interactions in more detail so that you can understand more of other therapeutic opportunities. I think while MSH3 and mutant Huntington lowering are clearly very important and are taking the first stage in terms of therapeutic opportunities, I think we should expand the stage like others have done in cancer to find multiple intervention points to manage risk that at one point this mechanism might be overwhelmed or they might not work so well. So in order to expand that, what we do in the lab is genetic screens by using this DNA scissors called CRISPR-Cas9. And we have engineered a line where we introduce this exon 1 with 118 CAGs. And we have shown that in wild-type cells, in blue, if you grow the cells in culture for 25 days, you have nice CAG repeat expansion. While if you knock out MSH3, that repeat expansion is stopped. And you can see those traces here on the right. In the wild-type cells that are Huntington, but with normal MSH3, you have expansion of the repeats, while in the MSH3 knockout, this expansion is completely stopped. So what we wanted to do is to see if we can find other genes that are involved in repeat expansion. And we done a screen of more than 800 DNA repair genes, and our readout was repeat expansion in this cell line. And what we find out through others that one gene called USP7, when deleted, leads to expansion of repeats. And interestingly, our collaborator Alex Sartori also find that the gene USP7 interacts with FAN1, one of the known modifiers. What USP7 actually does, it's a protein that puts ubiquitin on substrate proteins, removes ubiquitin from substrate proteins, and in this way can regulate the abundance of that protein in a cell. And what Alex and us nicely showed is that if you inhibit USP7, you increase FAN1 expression here. See here on the left, you see this black band. When you don't have USP7, you have more FAN1. And if you look at repeat expansion, when you delete USP7, you actually have increased repeat expansion as you have for FAN1. So if you lose FAN1 or if you lose USP7, you have increased repeat expansion. That is because USP7 controls the abundance of FAN1 in the cell. If you don't have USP7, you have less FAN1. And that being said, what we have shown is that USP7 actually binds to FAN1 and regulates its abundance in the cell. When you lose FAN1, you have higher repeat expansion. If you would inhibit this mechanism, you'd have more FAN1 and you slow repeat expansion. Interestingly, this USP7 also contributes to the pathogenesis of mutant Huntington, and this has been shown by Anna Pluchinek and colleagues in 2021, when they showed that actually USP7 interacts with mutant Huntington. So that's quite interesting and counterintuitive, because in their paper, they have shown that if you deplete USP7, you can rescue the pathogenicity associated with mutant Huntington in the cells. So USP7 is a two-edged sword in this case. It's a guardian hub in long-lived cells. Neurons are post-mitotic and must last a lifetime. And USP7 regulates multiple genome stability factors, including FAN1. It also controls protein turnover pathways for other important DNA repair genes, such as 53. And then for a cell that cannot survive, this is quite important to maintain these DNA repair proteins. But when you think about proteotoxicity versus DNA integrity, too much FAN1, you have fewer ubiquitinated substrates. And so you stabilize FAN1, which is good for the genome, but might be bad for accumulation of mutant Huntington. Of course, when evolution happens in the cell throughout generation, evolution didn't see Huntington's late onset is not part of the reproduction drive. So from a selective standpoint, balancing genome stability and protein stability until reproduction was the evolutionary pressure, and that was not to prevent Huntington at 50 repeats. So ESP7 has a dual role, maybe evolutionary compromise hub. In non-Huntington cells, they keep both DNA repair and proteostasis machinery flexible by turning ubiquitin dynamics. But what helps neurons survive in normal conditions becomes maladaptive in the presence of late onset repeat expansion mutations. So how would be this useful? I think one can now start to think about directed therapies. For example, you could use molecules that would glue USP7 to FAN1 to increase FAN1 abundance, and that should allow rescue of CAG repeat expansion in the cell. So with that, I want to finish, and I think the important thing for us and for me personally and our interaction with Sarah is to find new ways to attack both CAG expansion as well as protein aggregation. And while we're focusing on the DNA repair part, I think there's a lot of communication between DNA repair and accumulation of this mutant Huntington, and we should increase the genetic network so that we understand what's the best way to attack the disease. And with that, I would like to thank people that are involved in my lab, all the people in the Balmus Lab, our interaction with Sarah and Alex Sartori in Zurich, as well as the CHDI Foundation that is funding our lab quite generously, and some part of the USP7 work has been also funded by the Huntington Disease Foundation. And that I would like to thank you and then maybe pass to Sarah for her acknowledgements.
Thanks so much, Gabby. And I'd like to thank everyone in my lab. The slide's gone now, but basically everyone in my lab who does the work and all my collaborators and everyone I mentioned in my talk. Thank you so much.
Thank you both very much for fantastic talks. Really interesting, covered a lot of ground there. I'd encourage everyone who's listening, and there are lots of people listening and watching, to put any questions you might have into the Q&A. And then we'll try and get through some of those in the next 10 or 15 minutes or so before we finish. So first of all, and maybe Gabby, to start, you were just talking about the USP7, which sounds very interesting, perhaps something that people on the seminar are not so familiar with. So how do you think, you touched on it there at the end, but how do you think you might be able to manipulate that therapeutically? Like, what sort of approaches could you take?
So I think, there's a lot of work in gene therapies and approaching these by, ASOs and other modalities. But of course, these are very complicated therapeutics, take surgeries a lot of time. So I'm quite passionate about small molecules. And, one of the small molecules approaches that is more and more talked about is these small molecules that could glue two proteins together. So with the USP7, what you'd want to do is to allow the possibility that when USP7 comes close contact to FAN1, to have a molecule that glues it to FAN1. And what would then happen, USP7 would actually increase FAN1 levels in the cells, which is beneficial for the Huntington neurons.
Great, thank you. So a couple of questions maybe for either of you on MSH3. First, for Sarah, how did you establish the human tolerance of loss of function of MSH3?
Yeah, good. So we looked at the exome sequencing data, the worldwide exome sequencing data, the NOMAD database, and you can look at what you would expect for a loss of function, and what's observed, and a level of 0.6 of observed versus expected loss of function variance is said to be that gene you can actually live without. So, for example, Huntington, which is embryonic lethal if you knock it out, the expected observed is there is no one in the world walking around with zero, who's healthy or living, that is walking around with zero Huntington, because Huntington's is an essential protein in embryology. But for MSH3, there was, from the exome sequencing data, thousands of people walking around with either heterozygous MSH3 or no MSH3 who were well. So that is data that helps support that MSH3 loss of function is tolerated.
Thank you. So if you come to a clinical trial with an MSH3 therapeutic, either lowering or an inhibitor, how do you think, when you're setting up that trial, are there concerns that will be raised around cancer risk given its, its role in mismatch repair, irrespective of the fact you can't find variants? Do you think that is an issue when looking forward to these trials, which will hopefully
No,
be coming
I
through in
don't.
the next
Well,
year or
actually,
two?
I did at one point, but actually, I've been in those discussions already with the regulators. So I'm working to take forward the first therapy for MSH3, I think, which is going to be a gene therapy. And we hope to start that gene therapy trial next year, and it's going to be injecting gene therapy into the striatum to switch off MSH3. And so we've already had that conversation with the regulators. I thought that they would all be concerned about cancer risk, and actually they weren't. We presented the data about MSH3 being key in the pathogenesis of the disease, the genetics data showing that it's a very powerful modifier of disease progression. If you have less MSH3, your Huntington's progresses more slowly. And the regulator felt that Huntington's is such a terrible disease that a theoretical cancer risk was really not something they were worried about, because there is a predisposition if you have MSH3 double knockout, that you do have a predisposition to adenomatous polyposis coli in the colon. But those are quite rare variants, and we're not planning to knock out MSH3. So it's a very good question, and something I personally was very worried about. But when we actually presented to the regulators, what they're concerned about cancer was less. This was obviously a CNS only gene therapy and not something that was a small molecule and that is going to get to the colon. But I would say for a small molecule targeting MSH3, which Gabby and I are working on, and as many other people are, I think when it goes to clinical trials, I would suggest colon screening. And I think that's what we'll end up doing, is doing colon screening in MSH3 small molecule clinical trials.
And to add on that, Tom, if I may, one of the things that you look in cancer, and that's why I try to make that analogy with cancer, is that DNA repair genes, when they are lost, you have accumulation of mutations which drive evolution. And so when you lose mismatch repair genes such as MSH6 or MLH1, actually people like Serena and Xenial have created these cancer signatures, i.e. looking at clonal cancer and say, if you lose this gene, do you have increased mutation rates? And they can see that in MLH1 loss or MSH6 loss, you have an increased mutation rate. But when they look at MSH3, that mutation rate is almost not there. They couldn't distinguish an MSH3 knockout cells in terms of mutation accumulation from a wild type cell. So that again kind of saying that understanding the exact pathways and the redundancies in the system is really important. And what MSH3 is doing is really recognizing these hairpins. And that's one of the only thing that it does. So it doesn't affect the other parts of the pathway that are involved in cancer progression.
Yeah, thank you. That's very helpful. And actually, Gabby, maybe you want to say this one as well, which is related, which is on FAN1, DNA repair nuclease. And the question is, what would represent the optimal therapeutic strategy to enhance FAN1 activity safely in patients?
Yeah, I think that's a very good question, and it's something that we were thinking for a long time. The first thing that you have in mind as a DNA repair person, and not only when you think about nuclease, you think about scissors, right? So that cut DNA. So it's a little bit counterintuitive when you think, oh, what if you upregulate FAN1? You're going to start cutting DNA randomly. But actually, these proteins are tightly regulated in the cell, and that's why I kind of put all those genes in there that probably confused a lot of people. But they don't come alone. They actually are directed towards the proper sites by other proteins in the cell. So for example, FAN1, it interplays with PCNA, it interplays with other factors that bring it in the right location. So I think the first thing that you need to know is, the levels what you want to increase it. So I don't think you want to super upregulate FAN1. Probably you want to increase it half time more than the background. And there's some data out there from our colleagues showing that that might be a safe route. And actually, even in the papers that Sarah done in the past, it took us a long time to understand what the nuclease activity does, because when we were overexpressing FAN1, we couldn't see the requirement for the nuclease activity because having more FAN1 is good for the cells. And we never saw bad effects on the cell having found out. But I think the trick there is understanding what would be the therapeutic to increase it, 0.2 folds, 0.3 folds, and not 15 folds. Clearly, I think if you increase the 15 folds, will not be beneficial.
I agree.
Thank you. So understandably, quite a few questions on gene therapy, specifically and generally. So for Sarah, in terms of gene therapy approaches delivered directly into the brain through neurosurgery, do you think that this highly invasive delivery method will limit its application in clinic in terms of numbers of patients that could be treated and or not?
I think that's absolutely spot on question. And as Tom mentioned at the beginning, the gene therapy that I presented the results of last Wednesday was a really complex neurosurgery. And actually, Liam Gray, the professor of neurosurgery in Cardiff, went to the US to train in delivering the gene therapy. It's very complex surgery, 6 catheters, took between 12 and 20 hours. And there was one center in the UK doing it. I think when we started the study, that was the right approach. I think now we'd probably have a couple of centres with the neurosurgeons here at Queen Square and also in Cardiff. But the challenge is that neurosurgical procedure to deliver to the striatum takes a long time. And the challenge now is that neurosurgeons are working on shorter protocols. The gene therapy delivered to the brain in, say, 7 hours, but also better viral vectors are being developed. And we now have AAV9 children viral vectors, children of AAV9, that can be delivered with high potency in neurosurgical procedures that are between 3 and 5 hours. Because the viruses now are just much more potent, so lower doses can be given. Does that restrict gene therapy for the wider population? Yes. I think it just is in centers that can deliver that complex neurosurgery. Neurosurgery carries risks, but gene therapy is a single shot, it's permanent, and is a treatment for life. So I could talk a lot about gene therapy and the pros and cons, but is gene therapy and, for example, either MSH3 or Huntington the therapy for HD? Probably not. But I think what it does tell you is that the disease can be modified. That's what we didn't know before, and neurodegeneration can be slowed. And that gives us a real handle into developing more accessible drugs, oligonucleotides and small molecules.
Yeah, thank you. I completely agree with that. It's fantastic to see that the disease course and trajectory can be altered. I think that is
even in people with symptoms of the disease already. This is not people early in the disease. This was people with established symptoms.
Yeah. So there's a few questions around different modalities of therapeutic for targeting MSH3, and I suppose there's you could go from gene therapy to ASOs you might give intrathecally to small molecules that Gabby touched upon. I don't know whether you comment on which or whether a combination of those would be most useful, or perhaps we should develop all 3 for different situations.
I think Gabby's point about cancer treatment was really important. I do think for brain diseases like Huntington's and many other brain diseases that we work on in the DRI, combination therapy is going to be needed. And I think in Huntington's, it's going to be something targeting somatic expansion and something targeting toxicity. And so I think when you start developing therapies, the easiest one to start, well, it's not easy, but the first MSH3 therapy I think that's going to get into the clinic is going to be this gene therapy. It's going to be neurosurgically delivered to the striatum, and we hope to start that study later next year. I think after that, there will be oligonucleotides targeting MSH3 that get into the clinic, and also companies like Locus 23 have developed a small molecule targeting MSH3, which is hoping to go into healthy volunteers next year. So it's a busy area, and between the gene therapy, which is a single shot, versus the small molecule, which is obviously oral and can be given daily, we will then begin to assess the effect of MSH3 lowering. Now, when you start these sorts of studies, for example, gene therapy, we have to go into more symptomatic patients at the beginning because we can't start in stage 0 and 1 with a gene therapy for MSH3. So we'll see if we can slow the disease at when people have symptoms of the disease and then go backwards and eventually get to stage 0 and 1 prevention trials, which is where we want to be.
And one thing to add on that, if we would successfully make an MSH3 inhibitor for the field, while the mutant Huntington lowering will be specific for Huntington, MSH3 inhibition will probably cut across multiple repeat expansion disorders. So proof of concept of an MSH3 inhibitor in Huntington will probably open the avenue for that treatment in pre-symptomatic in many disorders, and will encourage people to think of the same kind of strategy of combination therapy with other, mutant proteins lowering, and really that will benefit the field.
And a follow-up question in the Q&A is, do the existing data on MSH3 indicate what level of knockdown you might need or reduction you might need in patients to modify disease? I think.
Yeah, so I am in the science translational medicine data that I showed, 42% lowering halved the rate of somatic expansion. So I think in the trials that we're going for about 50% lowering of MSH3 in the stratum, and I think similarly it'll be with small molecules in the body. Again, I think that 50% lowering will slow somatic expansion with just over 50%. I mean, I think in gene therapy we'll get more than that. It'll be about 80% lowering, but in the stratum. So, and I think with 80% lowering, you'll stop somatic expansion. But as Gabby showed, and in Steve's paper, the somatic expansion and the effects in medium spiny neurons is stochastic. It's not synchronous. Not every MSN is increasing their CAG at exactly the same time, and they're all dying at the same time. It's a stochastic process, and by targeting MSH3, we will be able to try and keep all neurons at CAGs below the toxic threshold of producing the toxic proteins.
And I don't know if you could put that into perspective for those on the seminar who maybe don't see patients, but if you lowered MSH3 by 50% and inhibited somatic expansion by maybe a similar amount, what impact would that have on someone with Huntington's disease?
It's a very good question. We don't know yet, but if you look at human genetics, our MSH3 polymorphism we found that was published in Davina's paper in 2017, it was about a 15% lowering of MSH3 in those people with the protective polymorphism. And that slowed progression across their lifetime. Remember, they're born with that polymorphism. So we know that that level slows the disease. But so we know it works. As I mentioned, David Altschuller's review, it's genetically validated target. Mother Nature's done the experiment. So I would hope by lowering 50 to 70% that we will be able to test in a clinical trial over 2 to 3 years that we're slowing this disease. And that's the key experiment that we need to do now.
And the other key experiment is correlate that with lowering with inhibition. We know small
yes.
For the protein abundance. So we need to know what is the amount of inhibition that kind of translates into the same. But it does seem that MSH3 activity and levels do go in parallel 1-to-1 ratio. So that's very encouraging, I think.
Yeah, absolutely.
And a clinical question. Given that, or a clinical trials question, given that the MSH3 targeting strategies you may want to apply or offer to patients in early stage 0 or stage 1 disease, and this is rather a big question, but can you share just some brief perspectives on how these clinical trials might run in terms of biomarkers? You touched on somatic expansion in blood and other biomarkers.
Absolutely.
Damage, and primary endpoints in the trial.
Yes, so that's exactly what I'm doing at the moment. I've been following this HD young adult cohort since 2017. Next year will be the 7th year of their follow-up, and we started the study in 2017. We recruited them, but next year the people who were recruited over 2 years will be coming back for their 4th visit, which will be 7 years for the whole cohort. And we now have biomarkers in stage 0 and 1, all molecular markers, CSF proencephalin, CSF neurofilament, brain imaging. And so I'm actually working very hard at the moment to raise £25 million, if anyone wants to give me £25 million, to do the world's first prevention study in stage 0 and 1. I've already discussed it with regulators, and we have biomarkers. It will run for 3 years, and that's what we're working towards. I hope to start that study in 2028.
That sounds fantastic. I'm aware that we're over the end of our time, but I've got a couple more questions. One was around the exon 1A, so slightly different. So we talked on at the beginning. I know that over the last 10, 15 years have been different views on the toxic species of mutant Huntington and their roles, particularly on exon 1A. But from your talk, Sarah, particularly, you sort of indicated that actually maybe there's a reasonable proportion of these toxic species, particularly when the repeat gets very long. So I wondered whether you wanted to comment on Huntington lowering strategies that do and don't involve targeting exon 1A, whether they both have a role at different stages of the disease, and so on. What are your thoughts on that?
I think it's a really good question. It's a question for an hour's answer, so I'll have to answer.
You can have a minute. There you go.
Have a minute. I personally think it's important to target both. Now that in medium spiny neurons and human neurons, we have now clearly shown using a digital droplet PCR assay that Mike Flower and Lucy developed in the lab, is that by the time you get to 140 repeats, 8%, you're producing this highly toxic exon 1 protein. Once you get to 170 repeats, it's about 20%. So it's occurring in human neurons. So I do think that targeting both are important. But the exon 1 is produced with longer repeats with somatic expansion. So it might be that in the shorter repeats that targeting full-length mutant Huntington and the N-terminal proteolytic species is enough. But I think one of the things with the therapy that we announced last week, it was targeting both. And in people with early symptoms, that clearly slowed disease. So moving forward, if I was going, as we develop therapies, I'm very keen on approaches that target both. So we can really target all the toxic species. That's moving forward, I think, is the right approach.
Great. Thank you. I think we're at the end of our time, which is a shame, because I think we could talk about this for many hours longer.
Really good questions.
But I'd like to thank both Sarah and Gabby.
So if there's someone else a question, I'm happy to receive it by email and respond. I know someone from Petr Cech's lab had a quite long and interesting question. Happy to answer that via email.
Yeah, if you've got more detailed questions, and there were a couple which I didn't read out, then please email Gabby directly or Sarah, depending on what the question is, and they would be delighted to answer you. Thanks to both of you.
I'm snowed under with emails at the moment because of last week, so I may not do it today.
Thanks to both of you for fantastic talks and discussions, and I'm just sorry we can't discuss for longer. Many thanks for all the people who've joined us, and this is a really exciting time for Huntington's disease. There are multiple ways in which we're trying to treat it, and it looks like we can treat it, and that brings hope to all of our patients, all of our families in clinics that we see around the UK and around the world. And it's thanks to the basic science of Sarah, Gabby, and many other scientists.
Lovely work that Tom has done.
Many other scientists around the world who have done over many years that's led us to this point. And I think it's it's testament to everyone's perseverance. And it's just great to be able to speak to people in clinic and offer them hope. So on that, I will say thank you very much and goodbye.
Thank you. All the best.